Hi, I'm Jan Witkowski. I'm Director of the Banbury Centre here at Cold Spring Harbour Laboratory, and we're here on the occasion of the 78th Cold Spring Harbour Laboratory Symposium on Quantitative Biology, which we generally refer to just as the Symposium. And the topic this year is immunity and tolerance. And we have here Jim Allison, formerly of Memorial Sloan Kettering, and now of MD Anderson in Houston, Texas. Jim was at the Banbury Centre in 2011 at a meeting on melanoma. And we were on tenterhooks during that meeting because it was when the FDA was about to pronounce on your antibody. Tell me a little bit about what your strategy was in devising this very effective antibody, and then we can talk about current status and so on.
Well, we devised the strategy early on. I mean, the idea was not particularly original. People have been trying to mobilize the immune system for decades because the immune system has incredible specificity for peptides that oughtn't to be in a cell. For example, those that are expressed in tumor cells or those that are generated by the mutations that cause cancer. This is what T cells see. The second thing is that there's memory, so once you get a T cell, it's there basically for the rest of your life. And finally, the immune system can adapt to changes in the tumor cell. Whereas other strategies for treating cancer have really none of those properties. They rely on taking something that'll kill cells, hopefully kill tumor cells more than normal cells. Lately, it's gotten more sophisticated with the advent of targeted therapies, which actually attack what's called the driver mutation that causes the cancer cell to be a cancer cell. But the experience so far is those can give short-term benefit in terms of shrinkage of tumors, but unless you kill every last tumor cell, what happens
is the tumor cells along with it.
And usually a lot of normal cells along with it, well, the tumor comes back and it's resistant to that drug, and then you got to find another drug. The immune system really offered the potential of doing that, providing memory where you don't have to necessarily kill every cell, you just keep them down. But in any event, these strategies hadn't worked very well for a number of reasons. But the one that we focused in on is that we really didn't know what you had to do to start an immune response. Everyone knew you need an antigen to give a signal to the antigen receptors, kind of like the ignition key in your car. It was pretty well accepted that you needed a co-stimulatory signal. There's a molecule called CD28 that had to be engaged at the same time. But people thought at the time in the mid-90s that the way you stopped responses, T cells just died, and that ended the response. But there was a molecule called CTLA4 that was very similar to CD28 that people began studying, and some people felt it was another CD28 molecule that gave a positive signal. We and a minority of people, on the other hand, felt it was an inhibitory molecule. And so we did a lot of experiments to show that was true. But as we were thinking about this, it occurred to me that maybe the reason that we weren't so good at enhancing T cell responses against tumors is we were hitting the on signals but ignoring the off signals, and that these things would kick in and protect the body by turning the immune system off. And so we had the idea of actually now, instead of treating the tumors, forgetting about the tumors and just treating the immune system and say, hey, whatever molecules are going to stop the immune response, we'll just suspend them for a while. And so we made antibodies to this molecule, CTLA4, and showed that when we injected them into animals, their immune system then would eradicate many different kinds of tumors. The mice were permanently immune, and it wasn't a problem. So we did a lot of work in this model and in this system in preclinical models, a lot of different kinds of tumors. As we would have predicted, there were two things about it that made me really excited and really focus on this for the next almost 2 decades now. And one of them is that, again, because you're treating the immune system and not the tumor, the kind of cancer is irrelevant. This has the potential to be a universal treatment for all kinds of cancer. And the second idea was if tumor cells are dying and initiating immune response, this approach could sustain that immune response But since almost every kind of tumor treatment kills tumor cells, they could be considered vaccines that would liberate antigens from tumor cells, and we could make radiation better, targeted therapies better, chemotherapies better, by bringing the memory to them. So you killed the tumor, and you can't kill every cell with your drug, and it goes away and it's gone. You get a good immune response, and it's there forever. And so,
I think you said about immunotherapy not having been very successful. I mean, I can have a fancy that immunological attacks on cancer have been going on for decades. I mean,
yes,
maybe since the early 20th century even.
And
they failed because they were going after the tumor cell rather than trying to manipulate the immune system.
Well, there were a lot of attempts to try to manipulate the immune system, but again, largely those focused on trying to activate the immune system to attack a particular thing,
right?
With a vaccine, a therapeutic vaccine, and so people would immunize, but what we learned when we studied CTLA-4 was every time you do that, you turn on CTLA-4 a little bit more, and so you're actually fighting against yourself by trying to just go for the on signal, as it calls it.
So 2 years ago now, the FDA approved it.
Right.
What's the experience been like in the clinic?
The experience has been tremendous in the clinic. Actually, there were in a phase 1 trial, which is typically a safety trial, where you just hope you do this after a lot of animal studies, whatever, and help try to identify whether there's a toxic dose and whether it's safe to use. And with most of these immunotherapy drugs, there is no maximum tolerated dose. I mean, with chemotherapy, with targeted therapies, you can get to a point where you make people really sick, and that doesn't seem to happen with immunotherapies. First of all, but the really surprising thing was in the first 14 people that were treated with just a single dose of this antibody, there were a few that showed objective responses already in the safety trial, including one woman that I had the luck to meet a couple of years ago. That was in the very first trial. She got a single injection of antibody. She had a grapefruit-sized metastatic melanoma in her lung, and after a single injection of antibody, it disappeared. And I happened to be lucky enough to be in Los Angeles when she was coming in for about the time she was getting her 10th year check. And I was a friend of her doctor's, and so he arranged for her to be there and to meet with me. And she, by then, that was 2 years ago, and so she's 12 years out from therapy now. She had a single treatment, no retreatment, no recurrence of disease, and if you could call it a cure, that's the cure.
And how many people have been treated now? You said that was the phase 1. What now that it's in clinical practice?
A lot of people have been treated with this because it took a rather unusual route to approval, because it was a completely new class of drugs. People were used to giving cytotoxics and look at them shrink tumors, and there are a lot of rules about how you evaluate. I mean, if you give a cytotoxic and the drug and the tumor grows or a new tumor appears, by definition it's resistant. But sometimes it takes the immune system a while to get going, and so everything was new, and it took people a little while to learn how to do it. But to date, and I'll give the total approximately as of today, and then maybe back up a minute, there have been about 18,000 people that have received treatment with this drug. The part of that was because of the positive responses. Medirax and Bristol Myers Squibb, the companies that actually developed the antibody, had what they call an open access trial, and they gave it to a lot of people that weren't necessarily on individual protocols. But the telling thing came in 2010, when the results of a very large phase 3 randomized trial, placebo-controlled trial, which is the most rigorous way of evaluating something, is you randomize all the patients, then you have a group of patients that don't get treated, and you see what happens. And so it took a long time to do this trial because the endpoint was survival. But two things were observed in that trial. First of all, the median, that is the 50% of the patients, was at about 4 months. So that was the first time in any randomized placebo-controlled trial, any drug whatsoever, had shown any survival benefit in metastatic melanoma. So that got a lot of attention. But the really interesting thing was that if you looked at the survival curve as it went out, by about 2 years it was 25%. By 2.5 years it was 23%. At 3 years it was 23%. At 3.5 years it was 23%. 4 years, anyway, after that it was flat. It didn't go to zero. By then the placebo control was basically non-existent. And so if you extrapolate from that to Sharon, who's 12 years out now, you're talking about durable benefit lasting a long time.
So the obvious question is, what is particular about that 20 to 23% that have this I think if I'd call it a complete cure over this period?
Yeah, we just don't know. I mean, I've got some ideas about it, but the bottom line is we really don't know. But what we know from our mouse studies is that if you add something on top of it that's the appropriate thing, if you study the mechanism, you can get that up from there, like add another agent, add another one.
Yeah, I mean, you mentioned that a bit earlier. So the function there is, or the mechanism is that other treatments like chemotherapy or radiation therapy kill off the tumors in the way that they do, but that increases the antigenic response? Or
yes. It dumps antigens from the dying cells into the immune system
and so with that sort of combination therapy, what rates are you seeing these days?
Well, I'm not sure. There's a large trial in prostate cancer with hormone ablation, but it hasn't been unblinded yet. It's a large trial that has finished. Some trials that are going to be reported next week, actually, although there's been some preliminary reports. It turns out that while CTLA-4 was the prototypical checkpoint, we call it, so this whole field is called checkpoint blockade now, that came from the CTLA-4 story. But there's another molecule that's very similar, although it works differently, called PD-1. And so last year, some phase 2 trials were reported where showing that PD-1 also had a high 20s response rate in a number of different tumor types. That was one of the things about CTLA-4. Melanoma was where the cancer in which the large trial was carried out, but there was really no scientific reason for that because there were also responses seen in the smaller trials in prostate cancer and lung cancer and ovarian cancer and many kinds of cancer, just as we would have predicted. PD-1, again, it's a different molecule, works differently. Another checkpoint, though. Last year's reported that antibodies to that could have, as I said, similar response rates. The durability isn't established yet because this has only been in the clinic for a couple of years, but there's no reason to assume that it wouldn't be the same. So we then went back to mice and combined the two antibodies and did all the control experiments, and it became clear, as we would have expected with molecules that have different mechanisms of action, they were at least additive.
Yes.
And so you get up to about 40% or so in the mice by using both. And that's kind of exciting, but next week at the American Society for Clinical Oncology meeting, there are going to be several results presented from a combination trial of anti-CTLA-4 plus anti-PD-1. And some of the preliminary data that's been released suggests that indeed in humans as well, it is the two together work better than either alone. And it's pretty significant because we're talking now about potentially curing not just a quarter, but somewhere above there. I can't put a number on it except as a guess. It might be 40% or something of patients with medicine. That's quite an exciting thing to go from with anti-CTLA-4, the first trials were in about 2000, 2001, to go in about 11 or 12 years to being able to cure about a quarter to maybe close to a half of patients with a disease which prior to that was uniformly lethal.
My recollection, and it may be from as long ago as 10 years or so, was that FDA was not at all keen on combination therapies, that in fact in particular that you couldn't combine two therapies that had shown little effect even though the combination might be better. I mean, that's not quite the situation here, but has FDA become more accepting
They,
of
they, they,
combination?
they seem to have, because there are combination trials running. I mean, it used to be that you couldn't, as you said, you could not use two things in combination unless each one had shown efficacy by itself. And you can think of ways, if you understand pathways, where that's just not going to be the case, where you have to have both in place to get a response. In fact, we showed that with NACTLA4 and a vaccine in mice, whereas neither one of them, neither the vaccine nor CTLA4 blockade, had any effect by itself. I mean, it was zero. You combine the two and you got 95 to 100% responses, and that's a true synergy where you took nothing and made it into a cure.
What are you doing to explore the, I guess the two sides of the same coin? On one hand, why that 23% responds so well, or actually, I mean, the response has been there, maintaining so well.
Yeah.
And on the other hand, why the other 75% don't do so well.
Yeah.
I mean, as a geneticist, I think we're going to be doing
Yeah.
genome sequencing to look for SNPs. And,
There's been a lot of genetic work. I'm not sure how extensive it's been, but people have ruled out polymorphisms in histocompatibility antigens, seem to have ruled out polymorphisms in cytokine genes or their promoters, polymorphisms and associations, I should say, with CTLA4 itself or CD28. So those things have all been ruled out. But nobody has done yet, and we're thinking about starting to do this now that we've got enough responders and non-responders. Do whole genome sequencing and see if anything jumps out and tells us.
Well, it's very exciting. So I hope we have another immunology symposium rather sooner than the last one we had.
Well, we're going to need one soon because this field is moving fast.
Thank you very much, Jim.
Thank you. It's been a pleasure.